Right, hello everyone, welcome back. So in this video we're going to be talking about protein chips. So with protein chips, everything is targeted. We're only looking for a specific set of proteins. Not all of the proteins in the world, but one we already decided we'll look for, in a sense. So you have these chips and they allow you to know how much of a protein is present and, well, whether it's present in the first place. So they're called like that because one of the components is always going to be a protein. So it really depends what type of chip you choose to use. You could use a chip where you have an antibody as the capturing agent, where you have a carbohydrate as the capturing agent. It could be whatever. I'll explain all these terms later. So here's how it's done. We'll take an example to make it clear, but I'll just count the definition part out of the way. Okay, so we'll take the allergy test so that we can count the how part out too. You already label a certain sample with color. So you take a sample from a patient, you label it with color. In our case, it's green. And if it's present, if that specific molecule is going to be present, then you'll have a green light showing that it's there. If not, you don't get the light. The green light I'm talking about is shown in the lecture notes in this really nice, neat image we got here with a bunch of dots. But how does it give you that green light? Well, if someone comes in and you're suspecting that they're showing symptoms of being allergic to something specific, you know that they're going to be making antibodies against the substance that they're allergic to. So it would be a good idea if you try and detect the presence of these antibodies. Now you can do that by having something called a capturing agent, or something that binds and captures the antibodies. If they're present, it'll detect them and bind to them. And there are so many capturing agents in the world. It doesn't necessarily have to be a protein too. The point is it should be able to capture the analyte and sort of like, how do I say this, and sort of like fix it on the array in the protein chip. Okay, it's a very specific interaction, and you're only going to get color if you get that specific reaction happening. The reaction between a capturing agent and an analyte will not, to give you that green light, will not happen unless the analyte is present. So once it's done, you get the green color, and it's that simple. There's always 4 things in the equation, with the first one, there's always 4 things in the equation, with the first one being the protein you want to detect. Okay, suddenly the pen stopped working. Anyway, the first one being the protein you want to detect, and then the second being the capturing agent itself, and the third one being the array itself, the array that everything happens on. In our case, this would be the green part here. And then with the 4th thing being the color that you're getting when things work out. In our case, it's green. Okay, so I would consider that as the how part being done. As for results interpretation and clinical application, let's go for results interpretation first. So in this image, you have 4 different rows, and what I mean by that is you can see how you don't just have one dot, you have multiple dots. And the only reason they're 4 is to allow you to really be sure that the first color shown in the first dot was 100% true. You don't want to make a mistake with this. You don't want a false positive. So you try your best to sort of get multiple testings done in the same protein chip. Okay, so I would consider results interpretation done with. Oh, wait, that's the wrong tick. Anyhow, let's look at clinical applications. Okay, so if you want to know, if you want to know if a cancer cell is aggressive or not, you can use a protein chip. It's not that you want to know if the patient has cancer. You already know they have cancer. It's that you want to know if you can easily get rid of the cancer cells. So the thing is, although this looks very complicated, if you pay attention, it gets a lot easier. In general, cancer cells can either be triple negative or estrogen positive. If you have a triple negative cancer cell, that's a very bad thing. I know it wasn't mentioned, but so I think that so as not to confuse you, it's important to just briefly go over what it means. It just means that the cancer cell doesn't have 3 certain receptors or 3 certain proteins. It doesn't express them. And these would be estrogen receptors, they would be progesterone receptors, and excess protein, HER2, I think. Which is actually a bad thing. It's a very, very bad thing. You don't need to know why or how. You just need to know that if it's triple negative, as in it tests negative for these 3 proteins, then that makes the cancer cell very hard to treat. On the other hand, if it tests positive for estrogen, then it means that the cancer cell would grow in response to the estrogen hormone. Ironically, even though this sounds like a bad thing, it's a good thing. And you're probably thinking, okay, how is that a good thing? It's good because it's much easier to treat than a triple negative breast cancer cell. So all you're doing is you're testing for these using suitable capturing agents, and you're seeing if the receptor is going to bind or if the protein is going to bind. And that's it, really. That gets us done with all the clinical applications. I'll see you in the next one.